speaker is Nicolas Salvy from University of Oxford.
So hello, my name is Nicolas Salvy. I'm a PhD student at INRIA in Paris, and I'm going to present work that was done with Tom Nichols while I was at the University of Oxford. It's entitled Morphometricity is Biased by Image Smoothness. Morphometricity is defined as the proportion of phenotypic variation that can be explained by macroscopic brain morphology. In our case, the phenotypes will be things like BMI and fluid IQ, and the macroscopic brain morphology is observed through MRI images of the brain. This is the equivalent of heritability, where we have brain data instead of genes. It's an interesting result because it summarizes the link between the high-dimensional brain data and the phenotype in one value. The framework we use to compute morphometricity is that of linear mixed effect models, and the first one relates Y, the phenotype, with X, the covariate, and Z, the standardized MRI images of the brain. So here, the morphometricity is the ratio between what can be explained by MRI images of the brain, so the variance of ZU, over the variance of the phenotypes once we have removed the effect of the covariates, so the variance of ZU plus the variance of the residual. A few years ago, morphometricity was computed for a bunch of phenotypes, and it yielded very high results. For instance, for BMI, morphometricity was estimated at more than 90%. This is what started our work, and what we try to do is understand why it got so high results. So first, let's look at how it is usually computed. The first linear mixed effect model I introduced is not convenient to fit, because Z, the brain data, is n times n, where n is the number of subjects, and m is the number of MRI data points, so usually higher than 100,000. So we would have to fit U with more than 100,000 components. Instead, we remove this annoying ZU part by computing its variance, which boils down to B sigma B squared, where B is the intersubject correlation of the brain data. So the first linear mixed effect model, which is now on the left, is equivalent to the one on the right, where we have replaced ZU with B, which has variance B sigma B squared, and now B is only n, of size n, and not m. We call the model on the left the generative model, because this is the one we'll use to simulate data, and the model on the right, the fitted model, because this is the one we'll use to compute morphometricity. Now, morphometricity is the ratio between the variance of B, so sigma B squared, over sigma B squared plus sigma E squared. This is true because Z is standardized, and so B is a correlation matrix. So this is how morphometricity is usually computed. So the question now is, what could have gone wrong? I told you that those two linear mixed effect models were equivalent, but for that, we need a key assumption. That is that we need to assume independence of the voxel-specific contributions. This is an assumption that comes from genetics, where this whole framework comes from, but in neuroimaging, it translates to the assumption that the contribution from one voxel is independent from that of the next voxel. So the assumption that things that are close together in the brain have independent effects on the phenotype, which seems a bit questionable. To test if this is where the problem comes from, we changed this hypothesis to incorporate the relationship between nearby voxels in the form of smoothness. So we're saying that a more believable assumption would be that the real U is a smoothed version of this independent U, so that voxels that are next to each other would have related effects. So formally, we say that U comes from an underlying stationary smooth noise process parameterized with smoothness F. And to take this into account, this is an assumption in the generative model, and to compute morphometricity, we need to take this into account in the fitted model, and we can do this by modifying the variance of B with the new BF instead of the brain relatedness matrix that we had before, where we replace the brain data with a smoothed version of the brain data. This is an operation that is very easy to compute. We don't have to compute any smoothing matrix, and this is very important because the smoothing matrix would be m times m, which is way too big. So now, to test if this was indeed where the problem came from, we did simulation. So for the simulation, we took real MRI images to have a real correlation structure. So we took gray matter VBM images from the UK Biobank. For 500 subjects, we had around 150,000 voxel values. We start by standardizing this Z so that the formulas are correct. And we simulated data with the generative model on the left by simulated epsilon and U 250 times for each scenario. So for each assumed parameter of smoothness. This got us a Y, so a phenotype. And then we estimated the smoothness with the fitted model on the left with the R package LMEkin. So in the first experiment, we simulate U under different hypotheses, from independence to increasing amounts of underlying smoothness. And we compute morphometricity in the standard way, so assuming independence of the contributions. What we can see is that as the model misspecification is higher and higher, we get an increasing positive bias. And this is the case even for very small values of smoothness. So here, the smoothness parameter is the full width at half maximum in millimeters, and the side of one voxel in our data is 2 millimeters. So 2 millimeters of smoothness is very, very small, and already we have a huge bias. And here, these are violin plots, so we see the repartition of results. So now we know that this hypothesis gets us a bias, but could we recover a true estimation of morphometricity with the BF we computed before? So here, U is simulated in the same way as in the previous slide, and this time we compute morphometricity by taking into account this covariant structure of U. And we can see with the white lines that we recover, on average, the true morphometricity. However, even for very small values of smoothness, so for instance 2 millimeters, the results are very unstable. So with 2 millimeters, we see that almost all the values are either very high or very low, but never around the average. So we could not trust a one-value estimate of morphometricity in this case. Okay, so now, finally, let's move on to real data. So the Z, the MRI images are the same. It's still VBM data from the UK Biobank. The Y of the phenotype of interest is the BMIs. This was the one that yielded very high results in previous works. And we include a bunch of covariates. We don't know what is the real underlying smoothness, but we can compute morphometricity by taking into account varying amounts of smoothness. With also on the left, when the smoothness is null, this is the standard hypothesis, so in the case of independence. And we reproduce the very high results from previous works. But as soon as we take into account even very small values of smoothness, morphometricity drastically drops. And so here, our data is consistent with a morphometricity somewhere between 0 and 93%, and we have no idea of where we are. So to conclude, when the latent effects of brain measures U are assumed independent, morphometricity is positively biased. This was the first simulation. Now the second simulation, if the true correlation structure is known, an unbiased morphometricity could be recovered, but it appears very unstable, and learning the true correlation on real data may not be practical. So all of these considerations suggest that morphometricity should not be used until new methods are developed that account for the impact of smoothness. I don't remember what is the number of my poster, and thank you for your attention.
Thank you, excellent talk. There's a few questions here. Someone's asking, considering that the U effects are conditional on other brain measurements, is the assumption of independence really unrealistic?
Well, it seems unrealistic because so compared to heritability, we have a relationship with everything around. This time we are 3D and not 2D, so everything is strongly related spatially. And yes, I would say that it's very strange to assume that the effects of two things next to each other in the brain are independent.
Yeah, this is too complex. I'm not sure what the person is asking. I'm not sure if you want, can ask him later. moving on to more questions. Actually, I have a question for you. I've played around with morphometricity before. You know, the variance component model has a very close relationship with like clonal regression, right? So then why not just use clonal regression directly to estimate the out of sample variance explained versus the morphometricity estimate?
Okay, I'm not that familiar with kernel regression.
Right, yeah. It's okay. If you're not familiar, we can talk later.
Yeah, let's talk later.
Discussing this, right? Yeah, okay. Not to the next one. There are more questions. Wait, do we have time for more questions? We can do more. We can do more. Okay, let's see. Could you maybe deal with the smoothness by working in the frequency domain somehow?
Maybe. So we tried a few things to take into account smoothness. We didn't really succeed. We haven't tried this. We could try this. Maybe this would be an interesting way to deal with it. I have no idea if it would work or not.
Does the voxel size matter? The collected value of each voxel is average or smooth version of raw values in your data.
I think the voxel size matters. If we have smaller voxels, then the assumption is even less likely because we see things that are even closer together, so more related. So it matters, and in our case, we had quite large voxel size, so I would say that it matters, and it's always bad.
Okay, let's thank the speaker again.
Thank you.